Hi, in previous session we have discussed different kinds of dendritic cells and macrophages. We learnt about their function, and we also learnt how these different kinds of dendritic cells play a very important role in various aspects of immunity. And the key point of that previous lecture is that dendritic cells are very important antigen presenting cells. Okay, the follicular dendritic cells, they play a very important role in B cell development and B cell mediated immune responses. And there is a one more type of dendritic cells, that is plasmacytoid dendritic cells, and these plasmacytoid dendritic cells plays a very important role in virus infection. We have also discussed macrophages, various kind of macrophages and its role, and finally we had discussed about how these reactive nitrogen species and oxygen species are generated, and I also gave you some idea about the importance of one of nitrogen species, that is nitric oxide, in non-immunological function. So, in this session we will take up two major cells, one is NK cell and another is NKT cells. Okay. So, NK cell and NKT cells, they are one of very important component of immunity, and basically these cells are present in blood, and this constitute about 5 to 10% of blood lymphocytes. Okay. And this cells has a packet of granules, and they are granular lymphocyte. They have some granularity. Okay, and these granules are very important for their function, as you can see in subsequent thing. Okay. So, these granules basically contain two very important set of molecules. One is perforin and another is this contain various kind of proteases. Okay, and we also call it as a granzymes. Okay, and this perforin and granzyme is very much essential in order to kill the target cells. So, when the NK cell get activated, then they basically forms a pore in target cells, and they deliver all these content of this granule, which is mainly consist of granzyme, and this granzyme is transferred into the target cell, and that will trigger apoptosis. Okay. And this the spore formation is mainly taken place by this protein known as perforin. Okay, as I told you in previous step. Basically, this NK cells are playing very important role or cytotoxic role, I will say cytotoxic role, to or against virally infected cells and tumor cells. Okay. So, they basically kill the virally infected cell and induce through apoptosis, and they also kill the tumor cell, as I have told you in some of previous session, that in our body there is always some transformation of cell, but that does not result to the tumor or cancer, right, because our immune system is intact and our immune system is keep on checking these kind of cells, and these cells basically express, if you remember my initial lectures, these cells keep on scanning, and then they scan for modified cells. Okay. If there is some modified cells, then these cells will come in action and basically kill those cells. Okay. So, the NK cells are playing very important role in against tumor cell. Okay. and they are also playing a very important role in virus infection. Please note that once the cells are infected by viruses, then there is no choice except to kill that cell. Okay, you cannot kill the virus inside the cell. The best way is you kill the cell, okay, the virally infected cells. So in that way you can prevent the virus spread in the host, and you can also restrict the number of viruses. Okay, if you kill the virally infected cell, you will reduce the virus. Okay, and that will also result to the reduction of viral replication. Otherwise, what will happen, when the cell are infected with virus, then after finishing the replication cycle, they will be liberated in outside the cell, and then these newly found viral particle will infect other healthy cells. Okay, in that way this viral infection will propagate. Okay, so NK cells plays a very important role, and how it plays a very important role? By a very unique and sophisticated mechanism, which we call it as a ADCC. Okay, ADCC is antibody dependent cell mediated cytotoxicity. Okay, so this needs an antibody. Okay, if we don't have antibody, the NK cell mediated killing will be not so efficient. Okay, so ADCCs are basically mediated by antigen specific antibodies. Okay, and there is a receptor for these antibody over the NK cells. Okay, and these antibodies basically bind with this receptor, and then that will trigger the ADCC, and this receptor is basically FC gamma R3. Okay, so basically the antibody which triggers this ADCC is IgG. It's a kind of antibody. There are several kinds of antibody. Okay, and this antibody basically binds with this receptor, and then it initiates ADCC. I have one nice animation which will help you in understanding this ADCC. Okay, so then the simple question arises that how this NK cell distinguish the correct cell and not correct cell. Okay, because the correct cells are normal cell, there we have all normal cell, and uncorrect cell is, it could be a virally infected cell or it could be tumor cell. So how this NK cell distinguishes? So basically in NK cell there is an activation receptor and there is an inhibitor receptor. So these activation and inhibitor receptor, basically there is some balance, and if this balance is disrupted, then this NK cell get activated. I will take all this these detail when I will take the viruses. Okay, so here just for your information, there could be a change in expression of MHC class 1 molecule, and this MHC class 1 molecule, if you remember my previous sessions, I have told you that this MHC class 1 molecule is present on all nucleated cells of the host. So if there is a reduction in number of this MHC class 1 molecule on target cell, then that will activate the NK cell. Another way is that there will be unusual profile of antigen MHC class 1 molecule. Particularly it is true for both virus and tumor cell. Okay, so these are the features which basically NK cell sense or screen, and if he finds if the cells find that there is a reduction in MHC molecule or there is an unusual profile of antigen and MHC class one molecule, then that will get activated and kill the target cells. Okay. So, NK cell get activated by interferon gamma. Besides this, there is a IL-2, IL-12 and IL-18. Okay. And this is a kind of signature of activated NK cells. Okay. And here I would like to tell that these NK cells are also used in some or other kind of therapeutic. Okay. And this is basically achieved by if you isolate the NK cells and treat with some cytokines, okay, cytokine or lymphokine, then they get activated. Here one example is that there is a cell known as LAK cells. Okay, this LAK cells are lymphokine activated killer cells, and this lymphokine activated killer cells basically are the NK cells are treated with IL-2, okay, and then this NK cells get activated, these natural killer cells get activated, okay, and this is used for some cancer therapy, okay. To best of my knowledge, this kind of cells are not generated in vivo condition. It's a ex vivo condition. We activate and then infuse and then we see some phenotype. Okay, this we also use in our experiments when we are using mice. Okay, so now there is a one simple question that how we can measure the NK cell activity, okay, whether they have a killing capacity or not. So we do one experiment which we called it as a chromium release assay, okay. This chromium release assay is a very simple experiment. Here we provide the target cells, we provide the target cells with some chromium salt. Here you can see that sodium chromate we provide, okay, or Na2CrO7. We put this molecule in the media. Over there this chromium is radio labeled, okay, and once it will taken up by the cell, this will be converted into the Na2CrO7, okay, and this cannot come out of the cell, okay, and when you put NK cells over there, then we will see there is a death of cell, target cell, and there will be a release of this radioactive chromium, okay, and in that way we can assess the NK cell activity, okay. So this is a one very simple experiment. This is a very gold standard experiment. However, these days we since you know that radioactivity has a lot of problem, we don't use radioactivity. So we have some another way. What we can do, we have a very sensitive assays for lactate dehydrogenase. So probably you know that lactate dehydrogenase is quite abundant in the cell, okay. So if the NK cell will kill the target cell, okay, then there will be a release of lactate dehydrogenase enzyme in the soup, and if we measure those lactate dehydrogenase enzyme amount by giving appropriate substrate or chromogenic substrate, then we can calculate the killing capacity of NK cells. So basically NK cells we can test the NK cell activity by these two ways. Now we don't use the chromium release assay, although this is a very good experiment, but we don't use. Now we use the lactate dehydrogenase because this anybody can do it, and you don't need a special facility, you don't need any special kind approval for those conducting those experiments, okay. So there are some factors which basically affect the NK cell activity, that is density and stability of antigen on the surface of target cells, okay, and antibody affinity. So, as you can understand, there is an involvement of antibody. So, if the affinity is weak, the NK cell activity will be not so good. Okay. Here I have a one short video which you can see how the NK cell works.
Often referred to as NK cells. These lymphocytes also possess Fc receptors on their surface, which bind to the antibody, leading to natural killer cell activation. The natural killer cell releases perforin, a protein similar to the membrane attack complex that inserts into the plasma membrane and causes target cell lysis. Natural killer cells also release granzymes, a group of proteins that initiate apoptosis. In some diseases caused by type 2
So, I hope you understand this ADCC phenomena very well. And now let us move on another very important cell type which we call it as a NKT cells. Okay. It is a natural killer T cells, and this natural killer T cells plays a very important role in recognizing non-conventional antigen. Okay. Basically, this NKT cells are a subset of T cells, and this subset of T cells plays a very important role against non-conventional antigen. Okay, and what is that? Before that, I will explain there are some properties. So, basically these NKT cells share function of both NK cell as well as T cell. They also share surface marker of both NK cell and T cells. Okay. So, NKT cells basically express T cell receptor that interact with that non-conventional antigen. Generally conventional antigen is protein antigen. Here it is a non-conventional antigen, and these non-conventional antigens are glycolipid antigen. As you know that the pathogen do have a lipid and lipid derivative, and these molecules also need to be taken care by the immunity. So, NKT cells are the key player against this non-conventional antigen or lipid derivative antigen. Okay. And all these lipid and lipid derivative antigens are present on variety of pathogens such as bacteria, protozoa, and they are also present in fungi. If I say bacteria, one of the very important bacteria is Mycobacterium tuberculosis. Okay. This Mycobacterium tuberculosis is, its outer covering is rich in variety of lipid and sugar derivatives. Okay. Probably you might heard the name of molecule known as LAM, lipoarabinomannan. So, these lipoarabinomannan are also presented along with one of MHC class 1 molecule derivative molecule, or it is similar to MHC class 1 molecule known as CD1D molecule. Here it is shown, and this lipid derivative molecules are presented to the NKT cell, and then there will be a activation of immune responses. Okay. So, overall the lipid antigen is presented by a family of molecule known as CD1D, which is similar to the MHC class 1 to the NKT cells. Okay. And then NKT cells get activated and induce the appropriate immune response. So, this NKT cell get activated during various infection, as I have explained you, microbial infection, and it is also get activated during inflammatory processes or in inflammation condition. Okay. And as I told you, when this NKT cell get activated, they produce a variety of immunomodulatory molecules, the cytokines. They can activate some population of immune cell and so forth, as I have explained you previously several times. They play a very important role against infectious diseases, autoimmune diseases, tumor, allergen. Okay, if there is some allergen will be there, NKT cell can activate and it will induce some immune response. So here I'm showing you in this schematic that how this NKT cells play a very important role in modulation of immune response. Here you can see that NKT cell present alpha galactosylceramide and lipid. Okay, this is basically a derivative of sugar and lipid, and this is presented to the NKT cells, and upon activation of NKT cell it can induce Th1 cytokine, for example interferon gamma, and or it can also induce Th2 immune response such as production of IL-4. So Th1 and Th2 response I will explain you in more detail in upcoming session. Here I will stop with this NK and NKT cells, and in next session I will take the last component of immune cell, that is B cells, T cells, and I will also discuss about one very important group of molecule or kind of nomenclature which is very important in immunity, which is very important to understand in immunity, known as cluster of differentiation. You are seeing lot of CD1 and CD various number, right? So we will discuss how this CD things came up and how it is helpful in understanding the immunology. Thank you.